We would like to invite all the speakers to sit here on the stage. Okay. So then they are ready for your question. If any, you can address question to specific speaker or to all the panel. It's up to you, and are they there? We will collect some question, and then they will answer all together.
Hi. I was just wondering about how things are going in the EU for translational research. I know that particularly Surat produced a lot of great science and also some things that potentially could become assays. But then there's additional things that need to be done in order to turn that into something that could be sent to validation. And I've been talking with a few people here, and in the U.S., the universities all pay a good deal of attention to IP, to intellectual property, and so they spin off companies. Professors leave and start up their own company, and apparently that doesn't happen very much in the EU. And so I'm wondering, how do you actually get from a university developing an assay to investigate biology or toxicology, and from there get to something which is actually ready to send around for ECVAM validation?
Unfortunately, in Europe, we do not have so much money. I mean, from the in toxicology, because if you see the European projects, even this Horizon 2020. So if you look for the word toxicity, you don't find very much. That is, in other countries. But I have a thought for the panel. I heard all the efforts concerning the alternative in vitro, in silico, and the structural activity and the read across. I wanted to know from the panel what they think, because to me sometimes it seems that the criticism on the weight, in the weight of evidence of all these tests is not too much, but the danger is that the results can be taken not to set priorities, but as an end word concerning the potential toxicity of a chemical. I don't know if you understood what I meant. So that there are good solutions to set priorities, but they should not be the end word. But some agencies, it seems to me that they are very happy with the what the in silico methods can show.
There is another one there.
Alexander Suvorov, University of Massachusetts. So there is a wonderful effort from both sides of Atlantic in the direction of walking away from animal experiments towards in silico and in vitro experiments, like toxicity testing. But we are not yet there, and toxicity testing still a lot relies on animal experiments. So I have a feeling, which may be wrong, that animals are left behind from the point of view that we still get from animal experiments very poor data, while high throughput methods allow us to get much more. So any rat or animal may be taken into thousands of small pieces, and every piece may provide genomic, metabolomic, transcriptomic, I don't know, DNA methylation, and etc. data. So my question is, what is your opinion about the fate of animal experiments during this transitional period? And if you can characterize any effort that is happening today to improve, like enhance animal experiments with modern methods? Thank you. And that is a question to all panelists.
So I think that you can start to answer, I would like to be a little bit provocative, saying, are the regulator ready to understand the meaning of these new frontiers in toxicology?
I'm always happy to start. Okay, I think first of all, there's a lot of cool stuff in all the laboratories in the US and in Europe. That's clear. The problem from my point of view is that this does not translate into regulatory sciences. We are lacking the transition because there is a very unattractive phase after I have published my method to make it applicable to a stage of standardization that it can actually enter validation. And even the validation process is suffering from less and less funding, so that there is very little incentives for academia to go into this. And we would have one only mechanism, which Harvey did refer to, which is going using market forces. And what do we do in our field? We penalize the methods which have IPR. We don't produce OECD guidelines on methods which have IPR, and the whole process is far too long that no company can survive the 10 years it takes until the method is going to be accepted. So we are creating an artificial gap which we could very easily overcome by allowing market forces. Somebody who has a good idea should have a patent, should make a lot of money, but we should have the method available very quickly in a standardized way elsewhere. And I think this is something we are definitely lacking. And the other part we are lacking is coordination. I said this already when I had the privilege to get our session started. We have in Europe a lack of institutions who are strategically planning, who are bringing things actively into the agencies, defining their needs. What in the US is called regulatory sciences, and which is flourishing at the moment, is not present here. Because the agencies are executing, the legislations are prescribing exactly what type of tests to do, and they're not open for interpretation in a way as US legislation is, which is hardly ever changed, but leaves the regulator the choice of methodologies.
Okay, so I was thinking about to start with the animals and the in vitro methods and alternative methods, and I think it's sort of it's a progress that is ongoing. So of course we have started to use the alternative methods and also in silico methods and so on as a screening tool. So it makes that you make a better selection for what you are really needing to test at the end. So I mean, you start to reduce the animals that are used. And I think it's a natural process that you gain then confidence, and you will be able to go a step towards a complete replacement. And then about the...
Can you use the microphone?
More close, you don't... Okay, sorry. And then I was thinking about this, what we are doing to trigger that the right methods are coming and how to stimulate this. Of course, it's a costly procedure, and we have heard that. But for example, within the ECVAM laboratory, we are now sort of doing these strategy papers. So we actually try to focus where we look for development, where you can sort of plug in a new method that is actually needed to make this battery of methods that is used for different endpoints. So I think that is a progress towards something that actually can be regulatory acceptable. Because you have, of course, to speak with a language that the regulators will accept, so you sort of construct sort of a planning that they can follow and that they can understand and feel confident to apply at the end.
Dr. Spielman has a response.
I'm sorry, I have to make a few statements that you may not like. Okay, Thomas also knows that in Europe we have the European Partnership for Alternatives to Animal Testing. It's an institution established between the European Commission and the major industry associations, which means pharma and chemical industry. And we have working groups. I'm a member of the supervisory board. And we have our 10th anniversary in the European Parliament. We are addressing, will be addressing the questions that you have indicated. So there is working groups just identifying developments, and I think Erwin Roggen, who is now in the room, can confirm that there's really the EPAA active working groups try to identify where are in vitro methods or new methods, non-animal methods that can be brought up to regulatory acceptance. So the regulators and industry and also ECVAM is collaborating. I have to say, every time I'm there, I say, why is EFSA not present? I have no idea. EFSA is the service of the Commission, may be independent, it should be also be there, because only translation is the job they have to do, okay? They are doing it, and you are invited. On the 1st of December this year is the annual conference, and it will be at the European Parliament, and there's members of Parliament present. And the point, of course, is where's the money for stimulating research? You may or may not know, I am one of the guys who had the idea of Seurat 1. And I think there is no Seurat 2 yet. It is not really honest to say the new 30 million project is Seurat 2. That's not true. And I'll be invited to talk at this meeting about the future of funding. There's been a big initiative against vivisection, you may have heard it, in Europe. So the Commission is under pressure to do more and will address this. In addition, a committee will be set up reviewing the outcomes of Framework 6 and 7 in order to stimulate research in this area. So the Commission is aware something will be done, and we all know it, this has not been mentioned, for IMI, the second IMI2 Innovative Medicines Initiative, the EU is spending 3 billion, and 30 million for this area is ridiculous, and I usually say, I've been involved in this, the Commission has spent 250 million, industry the same, for getting the in vitro methods for cosmetics accepted, so it was $500 million. If you want more complex areas like diseases, it should be in a similar range and not 30 million. So it's nice that there was 30 million, but it's ridiculous. And the last one is not invented here, what Americans don't like, not in NIH, not invented here. I am, you have to believe it, head of an advisory committee or supervisory committee of a company developing human on a chip, several organs working together, even 4, and for reasons that I don't know. The WIS never mentions us. And we are a spin-off of the Technical University Institute for Biotechnology in Berlin. You can find it on the internet, and I'm really unhappy that, again, a scientist from WIS, we always mention WIS, MIT and Harvard, we mention always others. For some reason, we invite them. When they are speaking up, they never do. And there was another good question, and this is my last one now. It's not published yet, but I can assure you, for nutritional purposes, our company has a contract finished with a pig on a chip. It is food additives, feed additive for pigs, funded by Novo Nordisk, so there's companies aware of that. We are on our way, and it is not as negative as you may have learned here. Thank you.
Right after this.
If I can go ahead and add a couple of things. So one of the really nice things about the United States government is that there is a mechanism that specifically supports the development of small businesses. So every single government agency has to set aside a certain amount of its funds. It has no choice in the matter, and those funds have to go out to support the development of small businesses. They are called Small Business Innovative Research Grants or Awards. Within the National Institute of Environmental Health Sciences, within the last 2 years, there have been awards to 15 small companies specifically focusing on the testing of chemicals for effects on differentiation of stem cells into differentiated populations, or for effects on differentiated cell populations. And so that is one mechanism that supports toxicology efforts within this particular arena. A second thing that is related to this is that there has been a revitalization of ICVAM, the Interagency Coordinating Committee for the Validation of Alternative Methods with the United States, with support from NICEDM, which is within the National Toxicology Program. And their focus is that a government agency, and in this case, most recently it's been coming out of the EPA, comes to NICEDM and says we specifically want to work in a specific area of regulatory interest, and in this case it's been endocrine disruptors. So there's been a major effort to try and develop in vitro alternatives that you've actually seen some traction on for the replacement of tier 1 in vivo alternatives like the eutatropic assay. So these are the things that are ongoing right now that I think moves the field along. But I think one of the things that's really critical is having very large institutional support for databases, because if all you do is support a database by a contract that only lasts 5 years, after 5 years, what happens to that database? And it needs to have a life of its own, and that's something that the U.S. government has been trying to support.
Thank you, Ray. I know Thomas has a quick
Yeah,
question.
I mean, as many of you know, I'm traveling between the worlds once per month between Europe and the US. And if I tell you what the biggest difference is, if I have a good idea in Europe, people will tell me, oh, we need a committee to discuss this. If I do the same in the US, they say, great, go for it. We cannot help you, but go for it. Yeah? And these type of committees Horst has been addressing all the time. We know for 13 years now that REACH needs integrated testing strategies. And we have one workshop after the other. But I see hardly any work being done. There's no work sponsored to produce ITS, and there's no ITS really actually introduced. That's the difference between the two cultures.
So are there other responses, please?
Perhaps I should say something after this. No, but I think there is actually a lot of things happening, and we have a good collaboration with the different agencies, and there is development ongoing. We have a lot of collaboration between between industry and also the governmental organizations. And I assume actually that the EU talks Risk 21 project is very much what was supposed to be the SURAT 2. It might be that it could be have been bigger, but honestly it's also sort of perhaps good not to have a too large project, but rather have collaboration in between different projects and initiatives, because giants might be a little bit difficult to handle. And for example, we have these other ToxMix projects that is looking very much on the same area of alternative methods on mixtures, just starting as well now. So that is another piece that is fitting into this net. And sometimes EU seems to be a little bit slow compared to other parts of the world, perhaps. But I think that is also because we try to anchor what we are doing in many places, and perhaps it's quite solid, the progress we are doing.
Thank you, Elizabeth. Are there other responses to these three questions?
Yeah.
Yes, please.
Sorry, if I can just share my experience. I'm French and I'm working in the US. So I think, as it has been said here, it's a big question of attitude towards what can you do with a discovery or so, a good idea you have. It's true that maybe as a caricatural French person, I would always say, okay, that's maybe too complicated. Let's think about it. And when I went to the VIS Institute, there was a lot of people were extremely enthusiastic in bringing a project to commercialization and translating it into industry. And I think that's one. The question of the attitude, but also the money is a big issue, and specifically, I mean, coming from the VIS Institute, which has been started with a big philanthropist, Mr. Vis gave like $250 million to start the institute, so it gives a lot of freedom to start new projects and commercialize things. And I think maybe in Europe it's less customary to have big philanthropy gift like that, and might be also another issue.
Right, we have, yes, please go ahead.
I think that in Europe, probably we have also, on a legislative point of view, something which is quite unique because we have a kind of schizophrenia. So some regulation, cosmetics, it is forbidden to use animal. Pesticide directive, the regulation for pharmaceutical compound, it is compulsory, and reaching something between. So I think that this also gives a picture and a big factor influencing all this diversity that we have in Europe. Not only committees, but also regulation are absolutely the opposite sometimes. Now there have been some questions related to in silico. In this case of in silico methods, I think this also is an advantage. It is somehow easier, for instance, to prolongate, to maintain, to sustain. You have seen maybe the logos within Vega. So year after year, project after project. Now we will start a UTOX 3, meaning 5 years more, plus 5 by default. So Vega will be maintained for 10 more years. To us, maintainment is not so costly as in other cases, because it's just virtual things. But in terms of acceptance, I think that regulators, they accept much more the use of this alternative method for prioritization. If you have to evaluate the individual single substance at this point, definitely you need a panel, a set of different tools in order to strongly support your final evaluation. In terms of the last point, transfer of the activities, indeed this is a critical point for in silico. As I said, it's easier. But now, hopefully, with Horizon, there will be much more attention to SMEs. So this also the possibility for transfer should increase. Some possibilities are also within the LIFE project. For instance, Brazil, we will apply the tools for the industry in dyes sector. So this is another possibility.
Thank you. I know Ellen has a response. Yeah.
Well, adding from the academic point who wants to contribute to these new concepts in Europe, so the top-down approach in the US really makes my work sponsored by actually the US and not by Europe. And I think it would be really helpful to have a little bit of top-down, from the regulators to we can contribute really to specific needs. So for me, it's very helpful how the US operates in that point of view.
I just was thinking about what Emilia was saying, and I agree with this. I think where we can actually gain funds and also expertise and improve a lot, it is to make it more horizontal. Both, I mean, from the scientific expertise from different fields in life science, as in between the regulatory network of different regulations in different fields. If you sort of get unified better, we will gain a lot. And there will then be, of course, more resources put all together.
We have one more response.
I just want to mention I didn't respond to the spin-off and culture in Europe, the difference. You may not know, I have worked for a US contract research institution called Battelle. So I'm a little bit trained in how research is being sold and spin-offs. And I have to assure that in Europe the culture is not that good. So as the example, as I say, this company Tissues had extremely difficulties after several rounds of funding by the ministries. And basically, since we are selling chips already, it's not even mentioned here, we are on the safe side. So there is a product already, and now it's getting better. So the culture is not that nice. And as far as taking into account what has been produced in the 6th and 7th framework program, the Commission wants to advertise in the area of funding small and medium enterprises, SMEs, funding in this particular area like humans on a chip. So but it is usually from the Commission coming. This is the biggest funding in the area. And if you think about differences in culture that Thomas says, there was a legal action. Number one, cosmetics directive, several times postponed, the 7th, and this was the initiative then to develop alternatives, funded again by the Commission. And it's similar with chemicals. So there is it's some difference, and it's not done by small companies that develop these things. And finally, a point that you may not see, human on a chip, there's differences. For example, I don't want to be too negative. The Chinese prefer a Chinese person on a chip, and that's where our market is. The Russians want a Russian on a chip. They don't want an American on a chip. May not be nice, but that's the way it is. And my company has signed a memorandum of understanding with the Chinese FDA, and you are not even aware of that.
All right, very good. So I think we have a question from the audience from our colleague from WHO. Yes, please.
It is on. It takes a while. Okay. Thank you very much. But now I have 3 points, if you allow me. Oh, sorry, Angelika Tritscher, World Health Organization. And indeed, the world is a little bit bigger than US and Europe. But I have 3 points now. First is that was triggered by Harvey's first comment and Thomas' reply with a spin-off and commercialization of ideas that come out of academia. So how do you see the issue with IP, intellectual property, and the open science and open data initiatives and the trend going on worldwide? Next point I have, we have a very distinguished panel. Each of you is involved in so many activities. How do you see that moving forward? There are so many initiatives going on. How can we better cooperate and collaborate and connect these initiatives to facilitate the uptake in the regulatory science and regulatory authority? And lastly, very practical, we heard a lot about very practical question about organ on chips and even people on chips, so to speak, of different ethnic background even. Just a practical question, because I'm not familiar, how difficult is the laboratory setup for this? So how quick can such methodologies, once they're really established, validated, actually be implemented? So very practical, how difficult is the laboratory setup for that? Thank you very much.
Very good. Who would like to start?
Okay. The science is global. The market is global. The only thing which is not global is regulation. And this is creating our problems. So we have to seek with the larger markets that we actually also create harmonization in the way we approach things. The organs on chip are a very nice example. These organs on chips are, or let's say these 3D constructs, which are the basis of this, as well as the platforms, are increasingly commercialized. But it's very difficult to get them across borders. It is extremely difficult to get skin construct and these type of things into parts of the world which do not produce them themselves. So we need to open this up, and we need the commercialization to actually make this happen. And I think IP, in principle, is a part of openness, because the IP is the contrary of a trade secret. You get your rights because you announce to the world how you do it. That's the patent. And for a short period of time, you can make money doing it. And I think this is the market mechanism, which is the only way of getting standardized, high-quality stuff available around the world, because some people see a business in this.
Any other responses? Please.
In case, I fully agree. In case of the in silico methods, the cooperation probably is easier. You have seen what the USA is doing with TOX21, TOXCASTR. So the data are available. Everybody here in Europe, in China. So this is also another part of the story of the picture. So it's also the beauty of the in silico part. It's not material. They are bit by software which go around. So data is one part of the story. Another part of the story is the protocols, so the same language. JRC is also for the OECD gathering and collecting the protocols used by the same or different, in order to compare the different methodology, in vitro methodology, because also for the omics, Thomas mentioned it. So this also is part of the story, to be sure that we speak the same language. And the software circulation, this is another part of the story. In our case, we are non-profit foundation, so all our software are freely available, and also the source code, if someone is interested, we can provide, and this be sure for regulatory bodies.
Ellen has a comment.
So Angelica, to your network question. So I think we need initiatives like we set up a couple of years ago. It's called ISTNet, International Stakeholder Network for Development and Non-Developmental Neurotoxicity, to get really the regulators, the industry and the basic scientists around one table and discuss how to go forward with certain projects. So, and we really had problems with going on and financing the next meeting after we've published our meeting report. But now I heard that EFSA seems to continue this and in collaboration with the OECD to really get this interconnection funded and the communication going, because I think this is the only way how we can get something to work when everybody sits around the table and discusses really what do we need and how do we get there. So at least for neurotoxicity, something's on the way.
No, I want to talk to discuss IP and also try to answer the other questions. IP for the company I'm talking about, or I'm representing to some extent, IP is key. It's the most important thing. Before they start working, they work on IP, and even before publishing, because the founder has successfully founded 2 other companies. So that's why he knows what is essential. So IP is key, and the Ministry of Research is supporting this, and also the technical university where the institution is. Coordinating at the EU. For a while, ECVAM did the coordination in collaboration with the OECD, at least for the test methods. And for some reason, it has depended very much on individuals and persons, as I indicated in my talk. I sent somebody, they said, if you want alternative methods accepted at the worldwide level, you have to send something. And I sent a guy, I paid him basically, I found money that he could work at the OECD for a year because there was no knowledge. And after that year, 4 methods were accepted. So there is ways of putting these institutions under pressure, and money and competent people is one point. And the other one, setting up labs. As I said, the company is selling already, and training people, so the setup, the basic setup, can be done in China. They will not do it with German tissue. There have been high-level people from the FDA in China. In Berlin, they have been trained, and they'll start the same in China, and it's done in other companies in Europe as well. So there is ways of transferring the technology.
Elizabeth?
Yes, I was just thinking a little bit about this with the regulation and international, that it's different, of course, and so on. But we have some, I mean, there is a global harmonized system for classification and labeling, for example. And that is, of course, looking at toxicity endpoints. Perhaps this sort of network should be in some way combined with a network that is developing new methods. And then you have already the international aspect of it. So perhaps there are connections without sort of starting new international foreign committees that should actually be combined in some sort of smarter way than they are today.
Thank you. And Ray?
We have made let me call it a small effort to try and expand the capabilities of TOX21. And the way we've done that is to make libraries of compounds available to other investigators, whether they be both in the private or the public session. So over the last 5 years, we've managed to set up, we call them material transfer agreements, with about 20 different investigators or organizations. The number of compounds have ranged from, say, 12 flame retardants up to 3,500 compounds, where somebody wanted to test those in zebrafish in a high throughput system. So we don't pay for it, but we do provide compounds that have been well characterized, where the data can be linked back to all the other data that's being generated in TOX21. So that's a small effort, but at least it allows for people to work off chemicals, because I'll tell you, one of the biggest problems I think most investigators run into is they might have a system, but they don't necessarily know what chemicals that should be tested in it, or even how to get the chemicals. And one of the things we found out in putting the 10K library together is even if you buy a chemical from a reputable source, sometimes the chemical is not the chemical you ordered. So it's a very small number, but out of the 10,000 compounds, there's at least 7 compounds that are highly pure, but they're not what we ordered. So you wouldn't know that unless you did the follow-up work to make sure you had, in fact, the right compound.
All right, very good. Well, we have a couple more questions from the audience. One right up here in front, please.
Thank you very much. Is it? No, it's on. Manfred Lützow from Switzerland, consultant in food and animal feed. I wanted to share with you some of the experience we had over the last almost 8 years with EFSA on read-across QSAR and using in vitro methods replacing toxicity testing. With a small exception of the QSAR, where there is a kind of standardized approach in areas like, example given, flavorings, where it's used as a routine screenings, the acceptance or non-acceptance has been unpredictable in the sense that it depends on the single expert or the single working group accepting a proposal or not accepting a proposal for read-across or for in vitro testing. And what I have not seen over the years is a response by EFSA of going across the panels, going across the evaluations and making proposals and putting forward a carrot that allows you really for applicants to go into that direction, to use these methods in a more consistent way. I was, of course, always impressed by the cosmetics approach, because there you have a legislation that demands it. I'm not much in favor of this kind of approach, but in a certain way, it is an example that shows you that the regulators have to put pressure onto the system in some way, whether it's a nice pressure or a negative pressure. You can discuss that, but I think it is necessary. Thank you.
Thank you. We'll collect another question or two before we start to the panel. Yes, please.
Hello. Yeah. Hi, I'm Maurice Whelan. I work at the JRC ACFA, my colleague of Elizabeth's. The last comment was very, very relevant. I think in the session yesterday on weighing evidence and uncertainty, one of the points we were discussing was as developers of alternative approaches, we need to be far better at identifying sources and describing sources of uncertainty to allow decision makers understand what's involved in terms of using alternative approaches. I just wanted, before I make a comment about the IP and so on, I feel obliged to correct some of the comments that were made. Europe, although we have huge challenges still to address and so much more to do, Europe has been and remains the world leader in terms of development and application of alternatives in the regulatory domain. And it's clear, for example, under REACH, and if you look at the reports about how the use of alternatives, although, as I say, major challenges remain, there's been significant evidence of uptake and use of alternative data. I mean, in terms of ITS, Thomas, I mean, to say that we're sitting around chatting about ITS and so on, out of the 11 case studies for skin sensitization that have been proposed at OECD for discussion, the vast majority of them have been developed in Europe. The project at OECD developing guidance on describing integrated approaches to testing and assessment, the project has been led by Europe, with vast majority of contributors by Europe. I mean, Horst, 10 projects at the OECD in developing new test guidelines are being led by Europe. Where are the other member countries? Where are the European member states? And I liked your comment about sending people there. But people are very reluctant to take on the challenge of leading projects at the OECD to bring these in vitro methods to the test guideline level. So, but just get back to the point about the IP. It's incorrect to say that an in vitro method which is protected by IP cannot be a test guideline. There's a provision at the OECD for performance-based test guidelines where performance standards are developed to allow in vitro methods of the similar class, a similar method, whatever, to be included all in a performance-based test guideline. So it's not right to say that and it would be a very dangerous message to give, because potential developers of in vitro methods might be put off developing in vitro methods for use in the regulatory domain if they understood that they couldn't protect their intellectual property. But what I wanted to come to was actually the point about this again around that point. In our engagement with little, small and medium enterprise companies and trade associations, IVTIP, for example, in Europe, which is a very dynamic group of small companies developing in vitro technologies, in particular engineered skin models and so on. And it was curious because their problem is not IP or issues like that. Their problem is actually getting credibility for the products that they're producing. How can they demonstrate the quality and utility of their products so that they can be used and taken up by end users in all types of sectors for all kinds of applications. And they were coming to us asking us to validate their methods for a particular regulatory endpoint, when in actual fact their intention was not to really address that market, to go after that market, but they saw the validation process as a way of giving some credence, some quality statement about what they were doing. And in fact, their intention would be to sell the skin model into the pharma companies or into the cosmetics companies, not for regulatory safety testing at all. So what it made me think about is that we really need to think about how can we address the issues of standards to encourage the development of biotechnology tools, in vitro models or whatever. And those standards can be used to really demonstrate the utility and fitness for purpose of these models, and to be able to compare them so that potential end users then can really use them with confidence and invest the money. Thank you.
Thank you. We'll accept one more question. Are there any from the website for our visitors that are online?
There are no questions as such, but what there has been is we've had several emails and comments on Twitter about the quality of the presentations and compliments to all of the presenters. There's been one recurring question about can they get hold of your presentations, and just to perhaps announce to everyone in here and out on the web stream is that we will be putting the web stream up on the website on demand within the next couple of weeks. And one of the things, just if I might add, we've had 900 people come to the conference, so in real time, and we've had almost the same number who've registered in the internet and on the web stream. So we've managed to double the presentation, the presence there.
Wonderful statistics. I have one question just to sort of bring in some of the biology. And we've been talking about wonderful things about IP, how to fund it, and how to get it translated across to regulatory agencies. But in terms of the biology of some of the platforms that you presented here today, I was wondering whether or not there was an interest in looking at developmental effects. Some of you addressed that. Ellen, I know you certainly did. But the idea is that right now there's a lot of interest because of a number of compounds coming down through the testing sequence that really require, it appears, developmental exposure and look for developmental effects. I was wondering whether or not the test methods that you spoke about today can address that issue of development, and at what stage during development does your test method actually emulate?
Actually, to this last point, because most of the best tissues at the moment come from stem cells, this is exactly the area where developmental aspects can be studied. And it's for good reason why we're looking at autistic cells at the moment and these type of things. So this is the big opportunity of the stem cell area in which we are. I think also it's important, US, Europe, this is not black and white. There's a lot of gray in between, but it is necessary sometimes to draw it a little bit black and white in order to make clear where the advantages and where disadvantages are, and how we can learn from each other and take advantage. For example, Europe is at the moment the pacemaker of legislation. It's the area in the world where new legislation is being made. Cosmetics, chemicals, pesticides, nothing is happening in comparison in the US. But technology-wise, there's a lot happening in the US. We see some programs which are second to none, which are producing at the moment regulatory data, a completely different approach to open access to data, which we could learn from on the European side. And I think what ECHA has been doing with the read-across and their read-across assessment framework is really advancing this field. It's the first framework to really make read-across something useful, to bring quality into this. And I think these are all very important developments where the other side will be benefiting.
Ellen has a response.
Yeah, actually, Bill, I would like to respond to your question. So development is really an issue of timing and processes. So, and I think the only way we can tackle this problem is really by creating batteries that take both into consideration. So at what time point what is happening at what time? And we actually, in one of the papers from this year, we proposed such a strategy for developmental neurotoxicity. You can distinguish the processes that that run during embryonic and fetal development are pretty much different from each other, and it's also depending on the cell type and what's going on when, where, and then brain region later come into the game. So it's I think if you consider all these different aspects, you have to come up with some sort of strategy that takes the different aspects into consideration. And if you think about it in a smart way, of course you come up with high content assays so that you can cover more than one endpoint with one assay. So this is something we're trying with the Neurosphere assay, but of course this is only a little part of the whole issue. Well, then you have to look at the rest of the body, of course.
Very good. I think first.
Okay, to make it very simple, the company is working on a mum on a chip, of course, but this is basically placenta. My background is developmental embryotox, and so I developed, you may know I didn't have the time to show, the so-called embryonic stem cell test, and there's a robot in preclinical testing at Roche that does this assay, so it can be done with mouse cells, and it has for in-house a fairly high predictivity. It's not accepted by regulators, but it is now being developed in Singapore and other parts of the world with human stem cells. So there is a way forward with stem cells and embryotoxicity. And finally, Alan is mentioning the other area is developmental neurotox, and the company in Germany is using or will be using Alan's human microspheres in a human on a chip. So there will be 3 endpoints, developmental ones covered by us.
Thank you. Any other comments from these questions? Okay, well, I think that we're at that point where we need to close the session. I want to thank, first of all, our speakers who did a fantastic job in delivering the information to us about so many important topics, and also, entering into the discussion, the great questions from the audience, and my co-host, of course, for setting up a wonderful session. So I thank you all very much, and enjoy the rest of the meeting and the close of the meeting. Thank you.